Okay, so some quick nomenclature here. So we've got, if we've got chromosomes where A and B are on the same chromosome, so they're linked, this is what we would call linkage. We could call that alleles capital A1 and capital A2, and capital B1 and capital B2. And the nomenclature for that, if you wrote that out, would be all of the letters or all of the genes that are on the same physical piece of DNA, A1 next to B1, so no punctuation between them, and then the slash, indicating that we're talking about two pieces of DNA, and then A2 and B2.
Okay. And now, where do we use the semicolon?
So, semicolon is, say we've got the same genes. So we've got A1 and A2.
Okay.
Oh, and let's go back, or I will go back and say that this is, we're going to call this chromosome 1, just for fun. And that indicates that we're only looking at one chromosome that both genes are on. If you want to use the semicolon, then we have to have two different chromosomes. So I'll just diagram those being two different sizes. So that's two copies of, say, chromosome 2.
Oh, okay.
So then we've got A1/A2, and then semicolon, indicating we're talking about a second chromosome.
Yes.
And then B1/B2.
Okay, so just to make sure, these
Yeah.
are homologous chromosomes.
Yes,
and
exactly.
And it's going to separate homologous chromosomes. That's when we use the slash.
Exactly right.
And when we talk about different chromosomes, we're going to use the semicolon.
Yep, so there you go. So there's, homology.
So the example
homology.
that we did today in class, we talked about homologous chromosomes.
Correct.
Okay.
Yes.
So it's the two copies of chromosome 1.
Of the same chromosome, yes,
Same.
exactly.
So,
the maternal and paternal.
I know we talked about this like
No, that's
the
fine.
first month, but
Yep,
I got confused again. When
Yes.
we're talking about ploidy,
yes.
and if we're talking about same chromosome 1, it's... Okay.
Yep.
Because, like, if this is chromosome 1, right? Would it be talking about this part, or this part and this part?
So, remember, ploidy is defined in the interphase.
Yes.
Before synthesis.
Mm-hmm.
So, pre-synthesis, you've got, you could consider this to be when egg meets sperm. They each are haploid. They each carry one copy of each chromosome. So here I'm diagramming an organism that only has one chromosome.
Yeah.
So that's going to produce a pre-synthesis organism that has two copies of that chromosome.
Oh, okay.
So that's the number of chromosomes it has is 1. It only has one type of chromosome. Therefore, its ploidy is defined by how many copies of each chromosome it has before synthesis.
So I'm talking about the part when it's tetraploid, right?
Yeah, so after
synthesis.
So after synthesis, then you're going to have what you just drew, like that.
Okay, so when we're talking about what we talked today, we were talking about one copy from dad and the other copy from mom.
Correct.
Oh, okay.
Yep. And here we have one copy from dad, one copy from mom, and then after synthesis, then, as you said, it's temporarily tetraploid.
All right.
Yep.
So these ones are homologous chromosomes.
Yep.
These are still homologous chromosomes.
Correct.
Okay.
Yes.
And then.
But we'll call them chromatids, technically
sister
speaking.
chromatids then?
Yeah. So these are, so all four of those, they're homologous. All homologous means is that they're the same
chromosome.
Yeah, they have the same genes in the same order, essentially.
Okay.
And Those are pairs of sister chromatids.
And sister chromatids, it means the homologous, two copies of the same
The same exact.
Same exact one.
Yeah, so
Okay.
paternal or maternal
Okay.
would be sister chromatids.
And that gets me to the next question that we were talking about.
Yep.
One of the assumptions
Yes.
that talk about the tetrad.
Yep.
Would it be only one, right?
So there's a yeah, this is a tetrad.
Oh, the whole thing is a tetrad?
Yep.
Oh, okay. All right.
Yeah. So all four of those sister chromatids together make up a tetrad.
And
Yep.
crossing over just happens once in the tetrad.
That's
one of the assumptions.
That's one of the assumptions that we
Okay.
make.
All right.
You throw a dart and it hits one of the
Like the
chromosomes
video.
someplace randomly, and yeah,
Okay.
that's where they break.
Thank you so much.
You're welcome.
You can
Absolutely.
ask, what do you need?
Sandwiches.
Another excellent question to bring up, which I should probably spend more time in class talking about, because
Well, my friend asked me in class. I was like, that's why I was like.
okay, so
Makes sense.
zoom in on a tetrad.
Mm-hmm.
What really happens, well, in cartoon form, what really happens is that there's a break made at the same location on two of the sister chromatids. And what you have to think about is that, I'm going to make this really obvious, this is going to be a 6 nucleotide long chromosome. And so we have two that are recently replicated. So these are, say, both chromosome 1 from dad, one from dad. Here's maternal. So the two sister chromatids from dad are identical. One's broken now and one is not. And mom's is going to differ at, say, a few nucleotides from dad's. But the two copies of her chromatid are going to be identical. Okay. So that break and drawing the X shape is connecting.
Okay.
The X is to make it obvious that there's exchange happening, that you start on one chromatid and that you move over to the other one. But really, that break is between the exact same two nucleotides. So it's not really like a big X-shaped thing that connects different parts of different chromosomes, which is kind of what I make it look like when I draw a big X like that. So it's really, let me make that a little bit more explicit. So when I draw something like this, that's really a break at the exact same spot on the two
So
chromatids.
all that part in the middle of the X basically disappears.
So it's a crossover between
Yeah,
two in the same two
it's a break.
nucleotides.
Oh,
Yeah,
that makes sense.
yeah. So there's no difference in the genetic material that results from crossing over. You get a chromatid that this little bit doesn't actually exist.
Exactly.
That's not new DNA or anything like that. That's just trying to graphically illustrate that the two pieces of chromosomes are connecting together. But there are no actual nucleotides there. And then you pick up the bottom part of that chromosome.
Okay.
So you would wind up getting, in the case that I drew here on the left, you would get an ATC there. And then it would connect to a GTT. So you'd get some of a paternal haplotype there. There's a difference there, where dad has A and mom has T. So we've got paternal on one end of the chromosome. But then on the other end of the chromosome, there's another SNP, single nucleotide polymorphism there. And so now that is a recombinant type gamete. It's got dad's haplotypes on the top end of the chromosome and mom's on the bottom.
Recombinant.
It's a recombinant type. Yep.
All right. Thank you.